resilience through diversity, biophysical heterogeneity protects physiological neurocircuit activity. Take it away, Scott.
Awesome. Thank you guys so much, Sue Ann and Milton, for the invitation. Apologies, I can't be there in person. Travel can sometimes be a hassle as a postdoc, but hopefully I'll be able to join you sometime in the future. And yeah, today what I'm going to talk about is sort of a convergence of my postdoctoral research towards this idea of understanding how biophysical heterogeneity protects physiological neurocircuit activity, with a little bit of a focus on how the mathematics helps us do this, given the aims of this conference. Again, please let me know if there are any issues, if slides aren't advancing, etc. So the first thing that we have to sort of have as a ground truth to dive into this is the fact that diversity is the norm in biology. Hopefully that's something that we can all appreciate if we look outside and we see the diversity of flora and fauna with our own two eyes. But something that's a little bit harder to appreciate is the fact that diversity is also the norm in our brains. And one of the easiest ways to notice this is if you look at the morphologies of individual neurons. So these are morphologies from human cortical pyramidal neurons taken as part of my postdoctoral research. And what you can really quickly see is that these morphologies are hugely diverse, even in the same cortical layer, which is often used as a sort of rough classification for these neurons. Similarly, classified neurons taken from this same cortical layer can have very diverse morphologies. And these diversities extend well beyond these morphologies, which are just easy to see, into these sort of baseline electrophysiological properties of these neurons that dictate their excitability, how likely they are to fire action potentials, and in turn communicate with one another. But there's still a question of what this diversity is doing. Is it a feature or a bug, a phenomena or an epiphenomena, meaningful or mere noise? Essentially, is this diversity that we know exists in our brain something that evolution selected for to serve a purpose, or is it just a side effect of what we know to be noisy and stochastic biological processes that lead to the development of these neurons, of these networks in the brain, etc.? What I'll hope to convince you of today is that there is a functional role for this diversity, and that there's strong preliminary evidence for this hypothesis in the fact that this diversity is lost in neuropathology, particularly epilepsy. Essentially taking a proof by contradiction approach, showing that by losing heterogeneity, we lose function in the brain. And the way I'll do this is by using computational neuroscience, and particularly some interesting mathematical tools from the field, to show you how we can connect from microscale features such as diversities and heterogeneities to mesoscale network dynamics, which are important when we think about function, which are important when we think about epilepsy, given that seizure, which is the hallmarking pathology of epilepsy, is a network level phenomena, hypersynchronous and hyperactive network dynamics. So the main work I'm going to talk about today was published recently in Cell Reports that again walks through how this reduction in heterogeneity might directly promote seizure-like dynamics in computational models. Three things I want to emphasize before I dive into this work is one, I think what you'll see throughout this talk is how the tools of computational neuroscience allow us to make linkages across sort of diverse fields within the field of neuroscience. So this work on its own is going to talk about how diverse neurons are in the brain, whether or not neuronal microcircuits are going to oscillate or not, and how both of these features are affected by pathology. And these are things that might be viewed as sort of different silos within the field of neuroscience, but can be connected very nicely using the tools of computational neuroscience. This is also work that is very interdisciplinary and collaborative. I very firmly believe that computational neuroscience is a uniquely interdisciplinary and collaborative field, where if you generate this cycle between experimental data and theoretical and computational modeling, you're able to find some really, really interesting collaborative results that you wouldn't have been able to otherwise. And so I just want to mention the amazing team that was part of this work. Dr. Valiente is one of my co-supervisors of my postdoctoral work. He is a neurosurgeon, so he is actually responsible for performing surgeries for patients with epilepsy, resecting live slices of human cortical tissue that allows us to do these experiments on human neurons. That tissue then goes to Dr. Homera Moradi-Chameh, who is an expert in the world of patch clamp electrophysiology and is able to perform these really challenging experiments to characterize the data from human neurons. And then Dr. Jeremy Lefebvre, who you might be familiar with since he's a more mathematically minded neuroscientist, one of my other co-supervisors, sort of motivates the math and the theory that undergird this. And then finally, I want to draw a little bit of attention to the types of models that we use in this work. And hopefully this is something that you're getting a little bit of a picture of from the wide array of talks that we've seen so far during this mini symposium. In the world of computational neuroscience, you can sort of choose neuron models that range across a scale from hugely biophysically detailed, apologies, I should have put my laser pointer on earlier, to a sort of balanced regime, the sort of the canonical Hodgkin-Huxley model, these conductance-based equations, where you still have biophysical detail in modeling individual ionic currents, but you're now idealizing things as a point neuron as opposed to thinking about these multi-compartment models using the cable equation to take into account this complex morphology. And then on the abstract side of the scale, you get things like the Poisson neuron model, which I'll use in this work, and I'll go through some of the equations to give you an idea of that. You might have this sort of initial reaction of, why would you use an abstract model, something that's so far removed from the biophysical reality, when we have these other tools, in particular when we have these intermediate tools like conductance-based neuronal modeling? And there's sort of two key reasons for that. One is that it works out so that the experimental data we had from our human cortical slices were directly constraining a variable that was better captured by a more idealized abstract model like the Poisson neuron, as opposed to these conductance-based models. And something that's one of my philosophies in computational neuroscience is hypothesis-based model generation, sort of choosing where you lie on this scale based upon what the underlying experimental data is and the types of questions you're aiming to answer. So we didn't want to shoehorn the data we had into a more complex model just for the sake of that complexity. But perhaps the more important reason is that using one of these abstract models preserves our ability to perform mathematical analysis, which again, I think will be of most interest to the audience that we have here at this conference. So I'll spend a little time walking through that. So what was the experimental data? What we found when we characterized these neurons from, again, live slices of human cortical tissue was reduced heterogeneity in a measure of excitability we termed a distance to threshold. This is just the difference between a neuron's resting membrane potential and its threshold voltage for action potential firing. And this heterogeneity was reduced in epileptogenic cortex, this red population here. Epileptogenic cortex, this is a clinically classified diagnosed region that is believed to be the initiator for seizure. And because it sort of is where seizures form and initiate and begin the spread throughout the brain, it's thought to be the most affected by the pathology of epilepsy. In contrast, when we compare this to two different control populations, one population that is from patients with epilepsy undergoing this resection, but this is tissue taken far enough away from the epileptogenic zone in order to get access to that it's deemed non-epileptogenic. It's seemingly normal, as healthy as we can possibly get, given limitations on human tissue resection. And then we also had another population taken from patients undergoing tumor resection, so no epilepsy anywhere at all. And again, you see that the coefficient of variation of this data is significantly reduced in the epileptogenic population as compared to both of our non-epileptogenic controls. Now, what we did with that data is design the Poisson neuron model where that distance to threshold had a direct analog with a shift in its activation function. And then we constrained the heterogeneity in those activation function shifts, which are randomly sampled from a normal distribution, to match the heterogeneity levels seen experimentally. So we chose these values of sigma E based upon what we saw in our epileptogenic or non-epileptogenic populations in our human data. We then connected this in an EI neuronal circuit. You've seen this mentioned a little bit throughout some of the other talks. So we have both recurrent connectivity between E cells and I cells, as well as intra-connectivity within the populations. This is a very commonly used sort of canonical idealization for a cortical microcircuit. And all we did was change these sigma values. We changed nothing in the network beyond the amount of heterogeneity in these sampled activation function shifts, and asked ourselves whether that change in heterogeneity alone would be sufficient to change the network dynamics and yield something akin to seizure. So when we have pathological or physiologically high levels of heterogeneity, what we notice is that the network dynamics scale more or less linearly with the drive to the system. So what you're seeing on top here in the background are raster plots where each dot represents a neuron spike. In the foreground, you see measures of this activity taken in moving time windows. Blue is a synchrony measure, black is excitatory firing rate, gray is inhibitory firing rate. And you see these measures averaged over 1,000 independent instantiations down below. And what you can hopefully quickly appreciate is that there is this linear increase in these measures corresponding with this drive to the system. There is, again, a more or less linear relationship in these dynamics relative to the drive. Contrast that with what we see, where, again, all we've done is decrease the amount of heterogeneity in the system. Now we see completely antithetical dynamics. We have this very sudden transition between sparse asynchronous firing and hyperactive hypersynchronous firing. And you can see that hypersynchrony by looking at these raster plots and these sort of very clear vertical bars. You can also see that via this increase in the synchrony measure quite suddenly, as well as the sudden increase in these firing rate measures. The fact that we see this sudden and abrupt transition into hypersynchronous and hyperactive dynamics is something we term a seizure-like transition. It obviously doesn't capture all of the complexity of what a seizure is, but it has sufficient and very strong parallels to what happens right at the onset in the initiation of seizure that we deem this a seizure-like phenomenon. Now, at this point, hopefully you're convinced that there is strong anecdotal evidence that there's this relationship between the amount of heterogeneity in the system and whether or not it will exhibit seizure-like dynamics. But this relationship is still at best anecdotal. We need the math to get into the guts and understand if there is a causal or a mechanistic explanation for this phenomenon. So what we did is we used tools from mean field reduction. We originally have essentially 1,000 differential equations, one for each neuron, where this is a relatively simple analog for a membrane potential. This just sort of has a linear relaxation, plus the synaptic connectivities, plus the various inputs to the system externally, and then just a little bit of noise. That gets passed into a sigmoidal activation function, and that sigmoidal activation function yields a probability of spiking at a given time window that gets passed into a Poisson process. So instead of thinking about 1,000 differential equations and 1,000 Poisson processes, we use mean field reduction to simplify this down into 2 differential equations, where these big U's now represent the mean membrane potential, the mean activity of our excitatory and our inhibitory population, and the big F's are convolutions of our little f's with this density function taking into account what the heterogeneity level is in these different systems. And what we find is that we can then perform linear stability analysis on this simplified system. And then again, now we have this nice simple matrix of a differential equation, which is something that you can use, very straightforward, grad level dynamical systems analysis on to do some stability analysis. So what happens when we do this stability analysis? Well, we find that there are 3 fixed points, and there's a bifurcation that occurs almost perfectly in this idealized system at the same level of drive is where we see this very stereotyped transition into the synchronous dynamics in the spiking network. So there seems to be, again, this very strong relationship between a bifurcation in our idealized system and the shift in dynamics in our spiking network. And if we dive into things a little bit deeper, what we recognize is that this is a saddle node bifurcation, where this yellow point is the saddle, the green is the node, the purple point is our unstable oscillator. And so as the saddle and node collide, yielding the saddle node bifurcation, we're left with just essentially a limit cycle that draws all of the potential solutions in parameter space. So if we're thinking about a single neuron that is oscillating, and this is what Eugene Zakevich was looking at in this cartoonification of things, that's what explains how a model of an individual neuron will exhibit oscillatory spiking. Within the context of our network, this explains how we transition into oscillatory network activity. So the math gives us a mechanistic explanation for this phenomena. This saddle node bifurcation only occurs when the heterogeneity in the system is sufficiently low, when the heterogeneity matches what we observed pathologically in our experimental data. Now, the math actually goes one step further in its utility. What I'm showing you here is a representation of one potential population of these sigmoidal activation curves in either our epileptogenic, less heterogeneous population, they're more clustered around 0, or the blue population, the non-epileptogenic, more heterogeneous, wider distribution. Something that we know mathematically is that when you have heterogeneity in these type of input-output functions, and you translate them into a population activation function, you get this linearization. This blue curve is a much more linearized curve relative to the sigmoidal shape you still see in this red curve. Now, what's interesting about this is this really nicely corresponds with something we found experimentally. So what I've plopped onto this figure here is what's called the FI curve. It's the relationship between an input current to the neuron and its repetitive firing frequency, Fi. And what we noticed in our data, very surprisingly, was that our epileptogenic neurons were actually less excitable in terms of this Fi curve compared to their non-epileptogenic counterparts. And this was something that was hugely perplexing, since we typically think of epilepsy as a disease of hyperexcitability. While this was confusing for us initially, the math helps to explain it. This is a population activation function, just like this in the background is a population activation function for our idealized set of neurons. So essentially, this linearizing capacity of heterogeneity in these population input-output functions helps to explain why you might see elevated activity in more heterogeneous populations of neurons at low levels of input, and vice versa for our pathologically homogenized set of neurons. What this means in terms of epilepsy is that we can reasonably achieve the network level phenomena of seizure, hyperactivity and hypersynchrony in a network of connected neurons without the individual units necessarily being hyperexcitable. A homogenized population of individual units may be sufficient in order to induce this hyperexcitability in the network level. Now, what I want to do with the last couple of minutes here is show you that these principles do generalize. So this is work that was recently accepted in PNAS and will hopefully be available for you all soon once we go through that long and arduous proofing process. But essentially what we asked ourselves is, can we look at a little bit more idealized system, a balanced neuronal circuit, and see whether the changes in heterogeneity have these effects that sort of vibe with what we saw in this more experimentally constrained network that I just showed you previously. And indeed, what we saw generally was that when the population was more heterogeneous, the activity over long timescales would be much more reliable and consistent, whereas the more homogenized system would be prone to these bouts of excitability that you can see in these traces, and you can also appreciate in these elements of increased firing or increased frequency over time in these heat maps, or the spectrograms, excuse me. You can also sort of appreciate this from the traces of an individual neuron. So essentially what's happening here is the red curve is this slow input, the slowly changing modulatory input to the system that is essentially checking to see whether the system is going to change its dynamics as this input changes. In a homogeneous system, this is prone to force the system into periods of high activity and then periods of quiescence. Whereas when things are more heterogeneous, then the individual neuronal units more closely track this modulatory input, yielding a much more consistent level of activity regardless of what this input is. And what we ended up concluding from this study is quite interesting, is that heterogeneity is actually serving to add a mechanism of homeostatic control to these networks. Essentially, when this term S0, which is the magnitude of that modulatory input, is very low, adding heterogeneity enriches network activity by increasing the spectral radius, essentially in allowing for there to be more activity, sort of more deviations that make for rich dynamics. In contrast, when this S0 is very high, and a homogeneous system is going to be very unstable, have this huge spectral radius, when you add in heterogeneity, you damp that back down and stabilize the dynamics back in a regime that is more consistent and reliable and predictable. So with that, I just want to give a thanks to my amazing team of postdoctoral mentors, both at the Krembil Brain Institute and currently at Sick Kids Research Institute. I recently made the transition across the street in Toronto for a next postdoc. I want to give a shout out to Dr. Murati Chameh, who does really, really amazing experimental work, without whom none of this human experimental data would be possible. The very sources of my own postdoctoral funding. And I'll mention that, I know I'm not there with you today, but I would always love to talk more about this work with anyone. Please reach out to me on Twitter, via my website, via my email, and I'd love to talk to you guys more about this work. And I'm happy to take any questions.
Thanks a lot, Scott. We've got time for a couple questions. Are there any questions from the audience? Yep.
So in the slide, when we show the mean experimental data for the mean activation function.
The mean activation function, yep.
So why is the epileptic one in general lower than non-epileptic?
Why is the epileptogenic lower than the non-epileptogenic?
It should go higher, like if it is sigma 1 after the like higher penetration.
Yeah, so, and again, I'm having a little trouble hearing you, so please correct me if I'm going off track of your actual question here. But what I heard was, why is the epileptogenic low here? We would expect that to be higher, right? Yeah. And so again, this was a very surprising experimental result. This is something that, again, this is a significant difference that's borne out. It's borne out in comparison to both of our non-epileptogenic populations. And again, it is something that is a little bit counterintuitive to our baseline understanding of epilepsy. But again, the way we were able to contextualize this is through this lens of homogeneity, heterogeneity, that a more homogeneous population of neurons is going to have a more sigmoidal, less linear input-output function. And so that explains why we get this lower level of activity at where this FI curve likely lies on these sigmoids, which is at the low level of input. So and the other thing I'll mention here is more from the experimental side of things. We did not perform any sort of perturbations or manipulations in order to make this an epileptogenic population. These are neurons directly taken from patients with epilepsy. Naturally occurring epilepsy is probably a little bit of a misnomer, but it's not something where we have a rodent model and we are adding kainate or, doing kindling or any of these manipulations to promote seizures. So what this says, at least in my interpretation, is that in human epilepsy, there may be pathways to the seizure state that don't necessarily rely on hyperexcitability at the level of single neurons. Most certainly that is a pathway to the seizure state. It's a pathway to the seizure state we've characterized very nicely in various, preclinical models. It's something that most certainly happens in a lot of human epilepsies, given the efficacy of our pharmaceutical interventions. But there is also the reality that the pharmaceutical interventions that we have for epilepsy that act more or less to decrease network activity, decrease excitability, only work for about 2 in 3 patients with epilepsy. And that's been a very, very hard ceiling for the efficacy of these drugs for basically half a century. So that implies that there are likely other pathways to epilepsy, some side doors to the seizure dynamic that may not be explained by the straightforward idea of neurons becoming hyperexcitable. We believe that this phenomena we've characterized as homogeneity is one of those potential side doors. And again, the way we can sort of see that is how this idea of homogeneity explains this admittedly very counterintuitive dynamic of hypoexcitability in these neurons. I will mention, if you take a look at the paper and the discussion, we talk a little bit about how this actually doesn't come completely out of left field, how there is evidence that neurons in the epileptogenic zone are actually hypoexcitable when in interictal periods when there isn't a seizure. So this is something that isn't completely out of left field as much as it is surprising at first glance.
Okay, cool. I think we're running into the break time. So with that, let's thank all the speakers.